20 July 2020, an alleged sexual assault was reported. The victim was Mary Wong, a female, and the suspect, John Smith, male. The traces about this case had been collected, as in this exhibit list. Through a series of operations, DNA extraction, DNA quantification, DNA amplification, and capillary electrophoresis, the questions about this case are able to be answered in source level, sub-source level, and provided as expert testimony.
The drink bottle was recovered from the scene of the alleged assault as a possible way to link the suspect to the scene. Analysis of the swab confirmed the presence of DNA and produced a partial DNA profile that resembles that of the suspect. The following likelihood ratios were calculated using allele frequencies, which is how often each variant of a gene is observed in a population, for the Australian Aboriginal, Asian, and European populations. However, only a partial DNA profile could be interpreted, due to a low amount of DNA being present. Therefore, it is inconclusive if the suspect is the source of the DNA on the drink bottle. The cigarette butt was also recovered from the scene to possibly link the suspect to the scene of the alleged attack. Analysis of the filter paper revealed the presence of DNA and a profile similar to that of the suspect. The following likelihood ratios were calculated for the Australian Aboriginal, Asian, and European populations. This provides very strong evidence for the prosecutor's proposition, and therefore the suspect cannot be excluded as the source of the DNA recovered from the cigarette butt. The victim alleged that the suspect used her mobile phone prior to the alleged assault. A mixed DNA profile consisting of at least 2 contributors was attained. Comparison with the reference samples provided by the suspect and the victim revealed that both individuals had likely contributed to the DNA profile. Therefore, it is 3,266,218 times more likely to observe this result if the suspect contributed the DNA, in addition to the victim, than if someone other than the suspect was the second contributor.
Hair and stain were found inside the victim's pants in the crotch area. The pants were treated with presumptive and confirmatory tests to identify the presence of semen. Analysis of the DNA profile from the cutout of the stain on the victim's pants revealed a profile consistent with that of the suspects. As displayed through the likelihood ratios of the Australian Aboriginal, Asian, and European populations, it can be observed that this DNA profile provides very strong evidence to the proposition that the suspect's DNA cannot be excluded as the stain on the victim's pants. Likewise, the recovered hair follicles produce a low-level DNA profile that appears to resemble the suspect. This DNA profile had rendered the appropriate likelihood ratios for the Australian Aboriginal, Asian, and European populations. However, due to the low amount of DNA being present, several allele dropouts were noted, resulting in only a partial profile to be interpreted. Therefore, it is inconclusive if the suspect was the contributor of the DNA from the hair sample. Upon visiting the sexual assault clinic, the victim consented to a vaginal swab to possibly reveal the presence of ejaculation. Analysis of the swab found that both the victim's and male DNA was present on the swab, to which additional examination revealed a DNA profile that was persistent to the suspect. Hence, due to the incomplete separation of the fractions, it is 1.43 times 10 to the power of 17 times more likely to observe this mixed DNA profile if the suspect and the victim were the source of the DNA in the male fraction, and 10,043,649 times as the source of the female fraction than if it were random members of the Australian population. This provides very strong evidence to the proposition that both the suspect's and victim's DNA cannot be excluded as the contributors on the vaginal swab. After the suspect's arrest, a shirt with a possible blood stain was recovered. The shirt was treated to presumptive and confirmatory tests to reveal that the stain was in fact blood. Examination of the shirt cutouts established a DNA profile that was consistent with that of the victim. Thus, as shown through the likelihood ratios of the Australian Aboriginal, Asian, and European populations, it's more likely to observe this DNA profile if the victim was the source of the DNA than if it were a random member of the Australian population. This provides very strong evidence to the proposition that the victim's DNA was on the suspect's shirt and suggests that the victim was injured at some stage, resulting in bleeding. However, it's outside the parameters of a forensic scientist's ability to comment on when or if the victim was injured during the alleged assault.
The results presented so far have been at the subsource or source level concerning the origin of the DNA recovered from each particular trace. These results do not, however, address the how or why the DNA is present, or the actions that led to the deposition of the DNA. Care needs to be taken when addressing activity level propositions with subsource or source level results, as the presence of a particular DNA profile does not explicitly confer an activity. Given this and the currently available case information, an expert opinion on the activity level propositions cannot confidently be provided. Therefore, it can be concluded that body fluids were likely present on each of the exhibits. These conclusions were made at the source and subsource level and provide very strong evidence to the prosecution's case, with the suspect's DNA likely to be present on the victim and their belongings, and the victim's DNA likely to be present on the suspect's shirt. However, an expert opinion cannot be provided on the propositions regarding the alleged activities or offences committed during the alleged assault, as it is outside the capabilities of a forensic scientist.